Apoptotic pathways. The mitochondrial intrinsic pathway. Mitochondria contain several proteins that are capable of inducing apoptosis, including cytochrome C. When mitochondrial membranes become permeable, cytochrome C leaks out into the cytoplasm, triggering caspase activation and apoptotic death. A family of more than 20 proteins, the prototype of which is BCL-2, controls the permeability of mitochondria. In healthy cells, BCL-2 and the related protein BCL-XL, which are produced in response to growth factors and other stimuli, maintain the integrity of mitochondrial membranes. When cells are deprived of growth factors and survival signals, they're exposed to agents that damage DNA or accumulate unacceptable amounts of misfolded proteins. A number of sensors are activated. These sensors are called BH3 proteins because they contain the third domain seen in BCL family proteins. They, in turn, shift this delicate life-sustaining balance in favor of pro-apoptotic back and backs. As a result, back and backs dimerize, insert into the mitochondrial membrane, and form channels through which cytochrome C and other mitochondrial proteins escape into the cytosol. After cytochrome C enters the cytosol, it, together with certain cofactors, activates caspase 9. The net result is the activation of a caspase cascade, ultimately leading to nuclear fragmentation and formation of apoptotic bodies. The death receptor, extrinsic pathway of apoptosis. Many cells express surface molecules, called death receptors, that trigger apoptosis. Most of these are members of the tumor necrosis factor receptor family, which contain in their cytoplasmic regions a conserved death domain, so named because it mediates interaction with other proteins involved in cell death. The prototypic death receptors are the type 1 TNF receptor and FAS. FAS ligand is a membrane protein expressed mainly on activated T lymphocytes. Where these T cells recognize FAS-expressing targets, FAS molecules are cross-linked by FAS-L and bind adapter proteins via the death domain. These then recruit and activate caspase 8, which in turn activates downstream caspases. The death receptor pathway is involved in the elimination of self-reactive lymphocytes and in the killing of target cells by some cytotoxic T lymphocytes that express FAS-L. In either pathway, after caspase 9 or caspase 8 is activated, it cleaves and thereby activates additional caspases that cleave numerous targets and ultimately activates enzymes that degrade the cell's proteins and nucleus. The end result is the characteristic cellular fragmentation of apoptosis. Clearance of apoptotic cells. Apoptotic cells and their fragments entice phagocytes by producing a number of eat-me signals. For example, in normal cells, phosphatidylserine is present on the inner leaflet of the plasma membrane, but in apoptotic cells, this phospholipid flips to the outer leaflet where it is recognized by tissue macrophages, leading to phagocytosis of the apoptotic cells that are dying by apoptosis and also secrete soluble factors that recruit phagocytes. The plasma membrane alterations and secreted proteins facilitate prompt clearance of the dead cells before the cells undergo membrane damage and release their contents. Numerous macrophage receptors have been shown to be involved in the binding and engulfment of apoptotic cells. The phagocytosis of apoptotic cells is so efficient that dead cells disappear without leaving a trace, and inflammation is virtually absent. Do not forget to like the video and subscribe to our YouTube channel.